that people say, oh, we humans can sense 5 basic tastes. And they are bitter, sweet, umami, salty, and sour. And you say, oh, yeah, but we can sense other things in our mouths, right? We can sense like the taste of oils. Why are they not taste? I am Dr. Fabiana Carvalho. I am a neuroscientist. So I've been studying perception for, I don't know, more than 25 years now. And we are going to talk about taste, but from a neuroscientific approach instead of from a sensory science approach. So we are going to understand how our sensory apparatus work and how the brain makes sense of stuff. Because, yeah, we have the sense organ, that's the tongue, but then it's the brain that perceives, okay? So the tongue is just a sensor, but the information coming into the brain must be integrated with information coming from the other senses, because nothing is perceived out of context. This is something we have to keep in mind, okay? So, okay, so those are the 5 external senses that we humans have. So we have vision, we have audition, we have somatosensation, that would be tactile, okay? We have gustation and we have olfaction. So we have vision, audition, and somatosensation classified as physical senses because they respond to physical information of the environment, whereas gustation and olfaction are chemical senses because they respond to chemical information from the environment, okay? And when I say physical information, I mean waves, okay? So we don't have yellow outside of us. We don't have a certain pitch outside of us. So those waves are transformed into sensations and perceptions. When a receptor is stimulated, we have an electrical impulse going to the brain, and then we perceive red, or we perceive this or that. And the same for the chemical senses. We have a molecule outside, C6H12O6, this is glucose. And when it binds our receptor, there is an electrical impulse going to the brain, and we perceive that as sweet. But the C6H12O6 in itself, in the environment, is not sweet. We cannot name it like that, okay? Right. Flavor is a construct. So, flavor is not a response of one sensory modality only, okay? So here we can have a look on how the sensory information interacts in order for us to perceive flavor, what we call flavor, okay? So when we talk about flavor, we have the senses that are stimulated once the food is in the mouth. So in the act of consumption, right? So we have gustation that will bring information about the basic tastes. We have oral somatosensation, that's the oral tactile sensation that will bring information about how hot or cold something is, the texture, and what we call chemical irritants, that will be the piquancy or pungency or numbness, okay? And texture, right? Yeah. We have the retronasal olfaction. So retronasal olfaction is when we put a food or drink in the mouth, and when we move it around, we are still releasing the volatiles there. So we have our mouths full of volatiles, and when we breathe out the air, when we do, we can smell. Okay, so this is retronasal olfaction. It's when the volatiles are pushed to the nasal cavity by the air when we are breathing out. We are doing this. And some authors also consider audition as the food eating sounds, as a consumatory sense, because the food eating sounds, they can hugely impact our perception of texture, okay? Right, so those senses here, they're called consumatory senses because they are stimulated when we're consuming, when the food or drink is already in the mouth. But flavor perception starts a little bit before that, okay? We do it with our eyes. So then we have the anticipatory senses. So those are the distal senses. So before consumption, we are looking at the food, we are smelling the food, we can listen to the food. So the sound of a sizzling steak, or if someone is eating a potato chip, I can listen to that, and this is already putting me in anticipatory mode regarding, in this case, texture, okay? And some authors also consider the haptic cues because I can touch the food, or I can use knife and fork or spoon or whatever to touch the food. And this is already giving me information regarding the texture, okay? So we have color, shape, so everything about vision, everything about audition, everything about haptic cues, and everything about aroma of the food as anticipatory senses. So before consumption, we are already in a very, very strong anticipatory mode, okay? Because of the anticipatory senses.
I'm curious about the haptic cues and audition, if they apply to the things surrounding the food as well. So
yes, they do.
Music, or like the plate that it's served in.
Yeah, yeah. So regarding consumption, we can divide the information as intrinsic sources of information. So when I'm talking about the food itself, the color of the food, the texture of the food, the sound of the food, but I can also talk about extrinsic factors. So it's everything that surrounds the food and is associated to the food somehow, like the servingware, packaging, environment of consumption, environment of purchase, logo types, you know, so everything goes in layers on the product itself, and it can affect the way you perceive the product per se. Yeah. Right. So we can consider these three consumatory senses here, so gustation, retronasal olfaction, and oral somatosensation, as the main senses for flavor. But as we have seen, all the senses participate in this construct that we call flavor, okay? And we are going to focus on gustation only, okay? Right. So what is a basic taste? Because we have heard that people say, oh, we humans can sense 5 basic tastes. And they are bitter, sweet, umami, salty, and sour. And you say, oh, yeah. But we can sense other things in our mouths, right? We can sense metallic, we can sense starchy, we can sense alkaline from carbonates. We can sense like the taste of oils. Why are they not taste? And why those are taste and those are not, cannot be labeled or called taste? The main reason is that taste is a formal scientific terminology. So the concept of taste can only be used if the sensation fulfills certain criteria, okay? And the two most important criteria are a receptor must be identified or located in a taste cell. So this is point number one. And the second point is this cell must generate a response, an electrical impulse in this case, and this electrical impulse must reach the brain area that is devoted to process taste information. So yeah, so these sensations, those oral sensations here, they fulfill. So the receptors have been identified and the information, the electrical impulse, finishes in this area we call primary gustatory cortex. So the electrical impulse will go to the primary gustatory cortex. So those are tastes. What about those ones? Well, some of them, the receptors have not been identified yet. Some of them are more than taste alone. Like the metallic, I don't know if you have tested, well, there is a course where we taste the iron sulfate with the nose blocked, and you cannot taste anything. But when you let go, you can smell it. The smell of iron, like, whoa. But we call taste because we mislocate to the tongue, but it's actually in the nose, okay? So those are not, some of them are almost there, such as the oleogustus, so it may become a basic taste in the future, and calcium maybe, but oleogustus is also tactile, yeah, because of the density or the viscosity of the oil. Okay. And this is just a little bit of an overview of taste discovery. Just for you to see how recent it is, the study of the chemical senses and eustachian. So the first ever receptor that has been discovered were the bitter receptors. So it's a family of receptors that we have. It's called T2R. We humans have around 25 different receptors for bitterness, okay? So they have located this family, the T2R, in 2000. So it was 24 years ago, okay? The first receptor. And we know about vision since mid-19th century was when Helmholtz came out with the theory of color perception. It's 150 years. And yeah, and it's still the one. So this guy could crack the code 150 years ago. So how advanced were the labs, were the vision labs? And the chemical senses were not studied at all. Only in philosophy, you know, the philosophers wondering themselves, this and that, but not really scientifically studied and measured and okay. So the first one was bitter in 2000, and then we have the sweet receptors being discovered in 2001, followed by umami in 2002, and then we have sour in 2006, and the last one was salty in 2010. Yeah, so actually what I'm going to talk to you is the very little that we know about how eustachian works. Okay, maybe in 5 years' time, I'll be back here saying, oh, do you remember that 5 years ago I told you that? Okay, no. Now they have figured out that's not really like that, okay? So what we know so far. Okay, so yeah, humans can sense 5 basic tastes up to 2024, okay? So this is the representation of the human tongue, and the surface of the human tongue is covered with a papilla, and in the papilla, I don't know if you can see those round structures here. So those are the taste buds, okay? And the taste buds, they are made out of taste cells. So we have taste cells composing a taste bud. And then we have a lot of taste buds in a papilla. And we have a lot of papillae on the surface of the tongue. So taste cell inside the taste bud, inside the papilla, on the surface of the tongue, okay? And papilla is one thing and taste bud is something else, okay? So you cannot see your taste buds. You can see your papillae, but not your taste buds. They are not seen from a naked eye. Right, so here we have the taste pore where the receptors are expressed. So those taste cells will express the taste receptor on its surface. So the molecules responsible for causing the taste sensation will bind there. Okay, so can I, yeah, just to show here. Okay, so this is the representation again of a taste bud. And each cell, not each cell, but yeah, we have a lot of cells here, more than 5, okay? We have dozens of them. But each cell will respond only to one basic taste. So we have the sweet cell, we have the sour cell, the bitter cell, and so on, okay? So one cell will express receptors for one basic taste only, okay? They are specific. And here we can see, like, for example, this orange cell here is the umami cell. So it's going to express this type of receptor here, okay, this 2,1R1, T1R3. And the main ligand, so the main tastant, the main molecule binding there is the glutamate, okay? So it will bind there and will cause an electrical impulse in the cell, and then this cell is connected to a neuron that has an axon. Do you know those fibers, long fibers? And this will carry the electrical impulse that will be generated in response to the receptor activation. So this electrical impulse to the brain area that will respond to umami. So you're going to see all the way up. And the same is true for all the other cells, okay? So the sourness perception is caused mainly by acids. So any substance that's able to ionize with the H+, so can cause the sensation of sourness. Saltiness, mainly sodium salts, but also lithium and potassium can cause the perception of saltiness. Sweet, we have a lot of molecules that can be perceived as sweet. The main ones are the monosaccharides and disaccharides like glucose or fructose or sucrose. They're all sweet. But we have certain sweet amino acids, for example. We have the artificial sweeteners too that can cause the sensation of sweet taste. And bitterness. Bitterness is a world in itself, okay? So we have around 1,000 bitter tastants that have been already identified. Actually, you can go online and search for bitter database. So you're going to see all the bitter compounds in there, and they can even tell you which receptor they bind to, okay? Yeah, so we have a lot of compounds, and as I said, we have around 25 bitter receptors, 25 distinct bitter receptors. So we have the bitter receptor number 1, and then number 2, number 3, number 4, number 5, and so on and so forth, okay? And we vary a lot genetically, which will impact the shape of our bitter receptors, okay? So depending on the genetic polymorphisms that we have, polymorphisms are expected genetic mutations in a population, okay? So we vary a lot. We vary in everything. If we compare the shape of our hands, the shape of our mouths, the lips, the nose, yeah, we vary a lot, and we vary internally too. And this is down to the receptor shape, okay? Okay, so depending on the shape of the receptor, this receptor can have a very high affinity to the molecule, so the molecule will bind and stay there. and then you're going to have an electrical impulse that's very strong, and then you're going to sense that it's very bitter, okay? A receptor can have a very weak affinity, so it doesn't bind well. So it stays on and off, on and off, on and off. So the message is going to be weak, and the bitterness perception will be weak too. And let's suppose you have a very strong receptor number 1, and I have a very weak receptor number 1. So for all the molecules that are going to bind there, for you they're going to be very bitter, whereas for me, I'm going to say, oh, this is not as bitter as you're talking about, you know? Yeah. But I can have a very strong receptor to certain compounds, which you will not have. And then you say, oh, to me, this is not bitter. It's okay. And I'm like dying, like, oh my God, this is so bitter. Okay? So we vary a lot on how we taste bitterness. So this is just a quick mention of that taste map on the tongue. So this doesn't exist, okay? One region of the tongue that specifically responds to one basic taste only, okay? Like the sides is only sourness, the back it's only bitterness. It doesn't work like that. But it doesn't work like that either, okay? People say, oh, you can taste all the basic taste across your whole tongue. Yes, that's true. But let's meet halfway. There is a difference in sensitivity, okay? Some areas of the tongue can be more responsive to certain basic tastes, but not exclusively responding to them, okay? So this is one thing and this is something else. This doesn't exist, okay? Papillae that are on the top of the tongue, they tend to be more responsive to sweet and salty than to sour and bitter. The diameter of the circle here is representing sensitivity, okay? And the papillae, which are in the back of the tongue, they tend to be more sensitive to bitter and sour than to salty and sweet. But there is the individual variance too. Okay, we are going to taste the basic taste after the talk, and you can see for yourselves where you are more sensitive to saltiness, to sourness, and so on and so forth, okay? Because as tasters, in addition to understanding the sensory apparatus, it's very important that we understand our own tongues, okay? So let's put ourselves into sensory tests and, yeah, learn about how we respond to things and how we can improve as tasters. There we go. Okay. So in addition to the individual differences in receptor shape due to genetic reasons, in addition to people being more or less sensitive to certain basic tastes on certain regions of the tongue, there is also the papillae density that can vary from one person to the other, okay? So here we can see individual variability of 2 people, okay? Person 1, person 2, across decades, okay? So here is a person of 20 years old that has a lot of papillae on the surface of the tongue. And this person here, not so much. And here we can see a 30-year-old. Again, a lot of papillae on the surface of the tongue, and here not so much. And we can keep doing this. Look. So like, this person at 80 years old has more papillae than this one at 40. Okay? So people can vary on that too. Yeah, there is a decay, yes. Okay, we are going to have less papillae on the surface of the tongue when we are 80 than when we were 20. But maybe at 80, we can have more than another 20-year-old person. Does it make sense? Hopefully, right? We are going to be this case. Yeah, right. So let's go into the, I think you asked about that, if there's something other than genetic that can change the way we respond to basic taste. So these are the cases that we have when we have a prolonged exposure to molecules. So this is true for any cells in the body that expresses receptors, okay? It can be, I don't know, not only receptors, but a cell that produces, it could be enzymes or antibodies in response to something that's exogenous, okay? So we have these two situations here. Sometimes when we expose the cell to the environmental molecule, we can observe a downregulation of receptors over time. So it's going to express less, and less, okay? But sometimes we observe the other way around. So the cell will express more receptors in response to that tastant in our case, okay? So which basic taste is which? Okay, so which ones we should avoid because we are going to become less sensitive, and which ones we should actually expose ourselves to them because we are going to get more sensitive. We are going to express more receptors in response to the stimulation. Okay, so the appetitive tastes, which are umami, sweet, and salty, are here. So the more we expose ourselves to excess, okay? Please don't cut off your sweets and your umami and your, no, please eat them. But do not exceed. So yeah, so when we expose ourselves to excess and prolonged exposition to those basic tastes here, we can see that the expression of receptors go down, okay? Whereas for sourness is the other way around. It seems that the more we are exposed to sour taste, the more sensitive we become to sourness, okay? And for bitter, there is a trend, but no effect, okay? So they didn't notice a response of the bitter cell regarding the expression of receptors after the cell is exposed to bitter tastants, and was quinino. I don't know how to say that in English, in this case. Okay? Right. Yeah. So this is the first stop in the brain, that is the primary gustatory cortex, or just taste cortex. It's easier. Okay. So all the axons of the neurons, they are connected to the taste cells, okay? So they are going to carry this electrical impulse, and those axons, which are tiny, tiny fibers, will be joined together, and then you will become nerves, okay? And then those nerves will carry the taste information, which is electrical impulses, to the brain. So all the way up till we reach the taste cortex. And within the taste cortex, we can even observe a certain brain areas within the taste cortex that preferentially will respond to a certain basic taste. So even in the brain, we can say we have the sour region or the sweet region, okay? So here's the average brain. So this is a functional magnetic resonance imaging. So those people were put in a brain scanner, okay? And they were given like tastants like that. So solutions with basic tastes. And they were mapping the brain areas that would participate in the perception of those basic tastes, okay? So we can calculate the average of response if we take into account all of those people, but we can look at their brains individually too and compare. So here we are looking for the variability, and we can see that we can have, okay? Like the sour area of this person is larger than this one. So this person, it was not tested here, but this person might be more sensitive to sourness than this person here, because you have more neurons participating in this response to the sour taste. Okay, so in addition to taste, when we are doing a sensory analysis of any drink and any food, the properties or the characteristics of the aftertaste is also very important, okay? So we have to evaluate the aftertaste in order to score a drink or food regarding the quality, okay? And we have different behavior, so to speak, of the aftertaste of the distinct basic tastes. So here we have the tastants for sweet, salty, sour, bitter, and umami. And here we have the duration of the aftertaste. So what the person would do is to like drink this tastant, spit it out, and then start paying attention to the sensation of taste is still lingering on the tongue, okay? And we can see that the longest one is umami, followed by bitter and sweet, followed by salty and sour, okay? So distinct durations of aftertaste. And even within one basic taste, so sourness here. If we are using tartaric acid or citric or malic or lactic acid, the perceived intensity and duration of the aftertaste can also change, okay? For the tartaric acid, it's like very quick, very sharp, right? So it's very strong but very quick, whereas the lactic, it's milder but longer. Okay, and then in between we have the citric and the malic acids. Okay, so yeah, this is just to bring now a problem, but okay, so let's go. So flavor is actually the integration of the information or the stimulation caused by volatile molecules and non-volatile molecules, right? So we say that the non-volatile molecules, they are in the liquid phase of coffee, for example. So we have a lot of types of molecules that will be trapped in the water, okay? And they will stay there. So yeah, we have organic acids, lipids, phenylenedines, melanoidins, blah, 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 blah. And those will be responsible for taste and mouthfeel, okay? Whereas the volatile compounds that we call volatile organic compounds are molecules that volatilize, so they float away. They don't get trapped in the water phase, okay, for any reasons. And so they are in the gas phase, and they are responsible for the aroma of the coffee, if I smell like that, or the retronasal smell, okay? If those non-volatile, if these volatile organic compounds reach my olfactory receptors retronasally, but they are all volatiles, okay? Right. The retronasal olfaction, this impacts a lot on how we perceive basic tastes. A lot. We are not really aware because we cannot separate what is a tongue stimulation or a nose stimulation. We just locate everything in the mouth, okay? Yeah, we don't, like, one example that I give is when we say hot taste. So this perception of piquancy is not taste, it is tactile. So it's a molecule that binds a tactile receptor, and the information reaches the tactile cortex. So it's tactile, but we as subjects, as perceivers, we describe that as taste just because it's happening in the mouth, okay? Yeah, it's the same with the retronasal olfaction, okay? Flavor is much more in the nose than it is on the tongue, okay? Right, so perception is active. The brain, do not forget what it already knows when we are exposed to something new, okay? So if you bring a cup with a liquid, a black liquid inside of that and put in front of me, my brain will recognize that as being coffee, okay? And it has a lot of expectations, okay, of what a coffee tastes like. And then when I drink the coffee, the brain will confirm, oh yeah, tick, 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 yeah, it is coffee. So what we do, what we call perception, is much closer to recognition. So we are much more monitoring the environment and comparing to what we already know, than actually sensing, getting the sensory information in, and evaluating the sensory information on its own. No, we are not doing that, okay? Life would be impossible if we were to do that, okay? So the brain learns from statistically sampling the environment. So I've seen how many water bottles in my whole life. Wow, thousands and thousands, I would say. So I have no problem whatsoever in perceiving this object as a water bottle. But I'm not perceiving, I am just recognizing or classifying or categorizing based on what I already know. So the brain, yeah, my eyes are scanning here, and I'm just classifying things. So this is that, based on what I know. And for flavor is exactly the same, okay? Yeah, so as we are exposed over and over and over and over, we build our internal models of stuff, of objects or events in the world, okay? So we all have internal models of what a table is, what a water bottle is, what a coffee is, whatever thing is, okay? And based on those internal models, we can recognize or categorize new stimulation in the world. Okay, so why I'm talking about that? So based on this statistical sampling of the environment, the brain starts to pick up on patterns. So things that coexist, okay? So this will go with this will go with that. Like we do that with colors and basic tastes. So we tend to pair red with sweet taste, because red fruits and red vegetables tends to be sweeter than the other ones, whereas we pair green, dark green with bitter, and light green with sour. Why? Because it's been like that. Yeah? So, yeah, we don't need to process like, oh my God, I was not expecting that at all. No, I was. Yeah. And one type of cross-model correspondence is called qualitative fusion. It's when it happens with taste and retronasal olfaction, okay? It's very strong. The brain will merge the two information. What the taste receptors are telling the brain and what the olfactory receptors are telling the brain. If they reach the brain more or less at the same time, if there is a coincidence, we are going to pair those two things. And this is so strong that there are articles showing that we need one exposition only to do that for life. And you cannot extinguish this memory. You cannot relearn. Yeah, it's powerful. So when we call this smell of caramel sweet, it really is, you know? Yeah, I've heard people saying, no, you cannot describe a smell as sweet or bitter. Oh, yes, you can. And you can because of that, because this qualitative fusion, this merging of information between retronasal olfaction and taste is very powerful. We cannot help ourselves of describing a smell as pungent or sweet or sour, and we can even go to colors. A smell can be dark, can be light, okay? Okay. But yeah, this is very, very, very important because perception is about the object unity. There's no such a thing as a caramel that's not sweet. So if we smell caramel in something, the brain will fill in the gaps and will find sweet there and will enhance it in order to fit the internal model. Okay? Yeah, the brain hallucinates a little bit. Not a little bit, a lot. Yeah. Okay, so those are examples of this qualitative fusion. So those are studies that were done in the 80s. So what they're doing here is to present people with water solutions, like those ones, without any tastant. So there's no tasting here, it's pure water, but then I'm going to put aroma. Those aromas won't taste of anything. It's water with aroma. And people are going to drink that. So those aromas will be perceived retronasally. Okay, so they are going to drink a water with almond smell or vanilla smell in them, and we can see that the perceived taste magnitude goes up. So they are describing that they are perceiving sweet taste, but there is no sweet taste there whatsoever. But the brain will fill in the gaps because there's no such a thing as a vanilla or a caramel that's not sweet. The brain cannot handle that, okay? So let's make it a little bit sweet. And here with a fruity aromas is exactly the same, okay? So this, oh my God, it's in Portuguese. But the sweet goes up, okay? Yeah, we can call this a gustatory illusion. So it's a retronasal smell that can cause us to perceive a taste that's not there. It's just like those optical illusions that we can see, like colors that are not there, or motion that's not there, okay? Right. And this article came out in 2020, where they actually measured the monosaccharide content in coffee. So they brewed coffee, they fractioned the brew, so yeah, fraction number 1, number 2, number 3, I think every 30 or 40 seconds, I cannot remember very well, but even so. So they got those fractions and they measured the total monosaccharide content in each one of those fractions and also the whole brew. And what they found is there are fructose, there are mannose, there are a lot of monosaccharides in coffee. But all of them, so this figure is the same as this one, but what we're showing here is the recognition threshold for sweet. So the concentration should be at least 3.5 milligram per ml, and they're all below. They are there, but they are not perceptible. We cannot perceive them. They're too low, okay? And then the authors ask themselves, if the monosaccharides, the simple sugars, are not responsible for the perceived sweetness, the natural question is, what does drive the perceived sweetness in coffee? Because we can describe a coffee as sweet. So what's going on? Okay, so when they looked at, okay, so they also had a sensory panel who would taste those fractions without knowing they are fractions. They were just samples. Okay, so those samples were put in front of them, they were tasting, and they would score how sweet, how intense was the floral flavor, how intense was the honey flavor, how intense was the fruity flavor for each one of those fractions or samples. And what they found is that the perceived sweet taste correlates with the perceived floral, honey, and fruity flavors. So the retronasal sweet volatiles are actually causing this. So yeah, if there is a caramel smell in coffee, there's no way that we're going to say that it's not sweet. Of course it is. Of course it is. Yeah. Yeah, I think that's the last one. Yeah. Thank you.